Good afternoon, guys. We start a lecture of microbiology. Our lecture today topic, we start a special microbiology, and today topic it's a human immunodeficient viruses, HIV viruses. For beginning, let's repeat what of these viruses. Viruses, it's unique microorganisms which don't have cellular structure. It contains only one type of nucleic acid, DNA or RNA, and have enveloped capsid, protein coat, or two types of envelope, protein coat capsid and supercapsid. Viruses obligatory intracellular parasites. It means that viruses cannot multiply without cells, without host cells, because viruses use resource of cells for synthesis self nucleic acid, for synthesis proteins, and the method of multiplication viruses calls replication. Then, now we start discuss one from the famous viruses, human immunodeficiency viruses, or HIV. But for beginning, we start from general characteristic family in which present these viruses. Family calls retroviridae, retroviruses. In this family present enveloped positive strand RNA viruses with unique morphology and means of replications. These viruses have unique enzymes which cause RNA-dependent DNA polymerase or reverse transcriptase. And these viruses replicate through the DNA intermediate. Then, DNA copy of viral genome produced by reverse transcriptase. This DNA copy integrate in the host chromosomes. It means in chromosome of host cell. Family retroviridae classify on three subfamily. Spumavirinae, in which present human foamiviruses. Oncorenavirinae, in which present human T lymphoma viruses 1, 2, and 5, and lentivirinae, in which present HIV viruses 1 and 2. On this slide present it's three of classifications, endogenous and exogenous retroviruses. That is lentiviruses, which classify, and also group of delta viruses, gamma retroviruses, spumaviruses, and beta retroviruses. A short about each subfamily. Subfamily Spumavirinae. These viruses don't cause clinical diseases. They produce characteristics vacuolated or foamy cytopathological effect. That's why the name of this Spumavirinae. Endogenous viruses have retroviruses sequences then integrate into genome. It's another group of viruses which sometimes describe. The presence of this virus is about 1% from chromosomes. Its ultimate parasites transmitted vertically. Also don't register diseases which may be caused these viruses. The next subfamily, oncoRNA virine, oncoviruses, another name. And in this subfamily present cancer-causing retroviruses, HTLV1 and HTLV2. They immortalize and transform target cells. Many oncoviruses express viral oncogenes, analogs of cellular growth controlling genes. And the last subfamily, which may be more interesting because today we discuss HIV, it's a subfamily lentivirine. In this subfamily present non-oncogenic cytocidal slowly viruses associated with neurological and immunosuppressive diseases. They cause slowly progressive chronic diseases, including AIDS. AIDS is acquired immunodeficiency syndrome, was primarily described as diseases for homosexual men, Haytens, heroin addicts, and hemophilics. They were known to be dying or normally being opportunistic infections. Several words about discovery. On May 1983, Montenegro in Paris, Institute of Pasteurs, published of descriptions of unknown viruses named LA1. In 1984, Gallo, in National Institute of Health, US, report to isolations of the human immunodeficiency viruses, HIV-1, from patients with lymphadenopathy and AIDS. Each group gave their viruses different names. But later, all start to call these viruses HIV. Then let's discuss the structure of these viruses. Retroviruses have spherical enveloped, about 80–120 nanometers in diameter, have capsid or course is cylindrical, surrounded by enveloped containing viral glycoproteins. Core or capsid morphology is used for classifying these viruses. The HIV virion core resembles as a truncated cone. Genome of retrovirus is also unique. It's a single-stranded, linear, positive sense RNAs, but diploid. It means two identical copies of RNA genomes. It's unusual for viruses. Also, in core, near the genomes present unique enzymes as called reverse transcriptase and other enzymes integrase, two cellular tRNAs, primers for reverse transcriptase, basic pair to each copy of the viral genome RNA. Also, in viruses present viral encoding protease is required for production infections vector. Then three main enzymes: reverse transcriptase, integrase, and protease. If we talk about structure of genome, then in genomes, retroviruses is franked with long terminal repeating LTR sequences containing promoters, enhancer, and gene sequences used for binding virion cellular transcription factors. Three main structural genes present in genomes: GAG, POL, and NV. The viral proteins are produced by proteolytic cleavage of the polyproteins by the virus encoding protease, then primary synthesis long polypeptide, which are later by viral protease cleavage on structural proteins. What proteins coding in structural genes? GAG, it's a short name, group-specific antigens, coding core and structural proteins. P24, it's a proteins of capsid. P17, matrix proteins. P7, nucleocapsid proteins. POL, from what? Polymerase, coding enzymes. P10, protease. P66 or P55, it's reverse transcriptase, and P32, it's enzyme integrase. NV, genes, from what? Enveloped. As a result, coding enveloped glycoproteins, GP120, and on this, and GP41 transmembrane proteins. Except structural genes, in retroviruses present regulatory or accessory genes. All of these genes coding different type of proteins. The name of these genes: TET, REF, NEF, VIF, VPU, VPR, and proteins P 14, 90, 27, 23, 15. And these regulatory genes and regulatory proteins, regulation strategic replications of viruses and host cells. For example, TET, transactivations are the viral and cellular genes transcriptions. REF, regulation RNA splicing and promotions export to cytoplasm RNA. VIF, promote of virion assembly. That's why it's all regulatory genes which produce regulatory proteins, which regulation strategic viruses and host cells. Let's discuss replications of viruses. Maybe all known, or if you don't know, then now you understand. Then HIV viruses or in first case infected immune cells. T lymphocyte, T helper cells, macrophage. That's why it's a tropic to receptors which are more often present on these cells. In first case, it's CD4 receptors of T helper cells, but also it may be chemokine receptors. For interaction with these receptors, on the surface of HIV viruses present glycoproteins 41 and glycoproteins 120. Glycoproteins 120, it's a receptors interactions between CD4 receptors, tissue tropism. glycoproteins 41 give ability to fusions membrane of viruses with membrane of host cell. That's why HIV viruses binds with CD4 receptors or another chemokine receptors, for example CXCR4. On the surface of T helper or macrophages. By glycoproteins 41, these viruses fuse with membrane of lymphocyte or macrophage. It initiates entry viruses in host cells. The result, genome of viruses with reverse transcriptase released into the cytoplasm. After this, reverse transcriptase start to synthesize DNA on the base of RNA genome of viruses. The first synthesis negative strand of DNA, then degrade RNA genomes, then synthesis complementary positive strands DNA, and as result formation proviruses, double-stranded DNA. These unusual transcriptions, it means when information rewrite from RNA to DNA, unique, and as a result, but really not usual for biology, for all life organisms. Result of this, it's about 5 areas per genomes when it's rewrite informations not from DNA to RNA and opposed, it means retro from RNA to DNA. Then very many mistakes during this rewrite informations. And it's the reason of instability of genetic of HIV. Very many variations of HIV, mutations, and variation of new strains, circulations in this population of viruses, and as a result, it means in humans. It's effective mechanism for escape of immune response. If it's very often changing genes and as a result changing antigens of viruses, that's why it gives ability to escape of recognition by immune system. Then these proviruses or double-stranded DNA integrate in host chromosomes. How? This double-stranded DNA transfers through the pores in nucleus and by viral enzymes integrates, splice into host chromosomes. You should understand not all DNA, circular DNA integrate in chromosomes. Another maybe remains in nucleus, in cytoplasm, and it's called non-integrated DNA, and it can be initiate cytopathogenic effect to destroy it of the cells. If it's big amounts of this non-integrated DNA accumulation in cells, that's why it can be initiate degradation of these cells. The integrated viral DNA transcribed as a cellular genes with host RNA polymerase. Then, if before this worked viral enzymes, after integrations, then information read as with host of humans DNA. And for this, viruses used host RNA polymerase. Full-length RNA is produced. For HIV, full-length RNA transcription process 2. That mRNA, ref mRNA, and vmRNA, GAG-POL mRNA, and GAG mRNA. The next step after transcription translations. Proteins translate from GAG, GAG-POL, and vmRNAs and synthesis long polypeptide. Then later this polypeptide be cleavage by protease. After this process, viral glycoproteins 120, 41, by endoplasmic reticulum and Golgi apparate migrate to membrane of host cells and integrations. GAG and GAG-POL polyproteins acylate and bind with plasma membrane containing enveloped glycoproteins. Then 2 copies of RNA associate in place various accumulations, proteins and glycoproteins. Then viruses envelopment and release by body. But another distinguished features in replication of these viruses that after release, the virion from the cells, viral protease cleaves the GAG and GAG-POL polyproteins later when virus is released from the cells. And only after this formate core or capsid in the middle of viruses. Then, if we assure to conclusion life cycle of HIV, then HIV binds to CD4 and chemokine receptors by fusions. Genome is reverse transcribed into DNA, and this DNA from cytoplasm integrate in nucleus and integrate in host cell DNA. Then, from these genomes, transcriptions and translation and synthesis viral proteins, viruses assembly and release from the cells by body. Another distinguished features of these viruses, then, it can be spread from one cell to another through the formation multinuclear gigant cells, which called syncytia. This process enhances the cytolytic activity of HIV. Why it's possible? Because if we return, then glycoproteins insert in membrane of cells. Then these glycoproteins can fuse with CD4 receptors on the surface of another cell, T lymphocyte or macrophage. That's why these two cells be fused and viruses can transfer from one cell to another without release. One of the problem of these infections that until this time people cannot completely to eliminate these viruses if from HIV patients. That is genetic divergence of these viral organisms. It means then viruses not identical but related genomes. The most of greatest divergence of localized in NV genes, which code in glycoproteins Y120 and glycoproteins 41. And this problem to complicate development of vaccines because viruses very fast to change the main glycoproteins against which formations antibody, yes, formations immune response. Usually these glycoproteins have a 5 variable regions. These regions can be changing, prolong the replications of viruses. In more important regions, V3, because against these regions, formations neutralizing antibody, formations main antibody which can be protected against these viruses. We discuss structure, replication of viruses. Let's discuss pathogenesis. HIV, as I said, tropic to immune cells. In the first case, it's a T cells with CD4 receptors, macrophages, and as a result, multiply and destroy of these cells. These HIV viruses induce immunosuppression or AIDS. Result of reductions of number of CD4 T cells, it's dissemination of T helper functions of immune response and delayed type hypersensitivity. Function of CD4 cells or T helper cells, its regulations main chain of immune response, adaptive immune response, and T helper regulation, and cellular immune response, and humoral immune response. Also, these viruses are infected macrophage, and macrophage is the main cells for activations adaptive immune response, and the main cells, it's phagocyte of innate immunity. That's why initial infections of macrophage with M-tropic HIV strains by interaction with CD4 CCR5 receptors and macrophage lineage cells become to persistently infections. Circulating macrophages, microglia cells of the brain, pulmonary alveolar macrophages, dendrit cells and other cells of monocyte macrophages lineage spread the viruses and contribute to the clinical manifestation of the cells. In other words, these cells to spread and transfer viruses from in different organs and tissues. And later, T-cell tropic HIV strains involve in infection. On this picture, you can see T lymphocyte, usual form, and HIV infected T lymphocytes, in which, yes, destroyed of receptors, and as we can see, change in morphology of these cells. And these cells cannot be worked in immune response. Then, these viruses released from the cells usually by body. It means that is don't destroy it immediately or don't have directly cytopathic effect. But these viruses destroy cells by another mechanisms and earlier or later infect these cells to death. How HIV viruses destroyed of immune cells? The first, and we said about this, an accumulation of non-integrated cellular DNA copies in viral genomes, which initiate cytolysis of the cell. The next, increased permeability of plasma membrane, because when virus is released by body, it's destroyed membrane of the host cells. As a result, not immediately but later, this causes change in permeability of membrane, change in osmotic pressure, and lysis of the cell. Third, syncytia formations. One cell fuse with another cells, and result of this, very many immune cells fuse in one B cells and as a result don't work effective against HIV viruses and against another type of infection. And as most of viral infections, HIV can initiate apoptosis, genetic death of infected cells. Ability to HIV to kill the target cells correlate with the amount of CD4 cell molecules which is expressed on by cells. On this slide we to join, yes, pathogenic mechanism of HIV infections. Target cells, T lymphocyte or macrophage, persistence infections, productive, then it's a sum of period, yes, persistence or latent period. Then, but earlier or later, it's cause productive infections, immunosuppressions, and as a result, opportunistic infections, or if it's macrophage, that it's usually brain diseases and neuropsychiatric manifestations. Then, how to correlations amount of CD T cells and diseases. The first, HIV infections associated with the boost of the virus productions and viremia. It's in first period during the first month, maybe, and it's the period of viremia initiate of mononucleosis-like syndrome or flu-like syndrome. Then, as it starts on initial, it's a normally healthy immune cell, then it's try to decrease the number of viruses, and viruses levels in blood really decrease, but viruses survive and to continuous multiply in lymph nodes. Symptoms to lack. Yes, people usually may long period don't know then an HIV infection. It means to start clinically later periods. It may be 1 year, maybe several years, but viruses don't eliminate, it continues multiply. Level of CD4 T cells significantly decreased. The structure of lymph nodes destroyed and patients earlier or later becomes to immunosuppression. On this you can see how it's the first viremia and decreased number of viruses, but viruses survive. Then CD4 T cells in beginning also maybe sometimes more synthesis, but as this virus is continuously multiplying lymph nodes, the level of T cells more and more decreased. And as the level of T lymphocyte critical to down, HIV viruses start active multiply. It means period of when latent HIV infections transfer to AIDS, acute immunodeficiency syndromes. Also in this picture you can see how to start to formate antibody against HIV. Antibody try to protect against viruses, but it's less effective, no effective. Then, by reductions of number of CD4 cells. Some of these reasons we discussed. HIV induce cytolysis of these cells, cytotoxic T cells to destroy T infecting cells, natural terminal differentiations of T cells due to the response of large HIV antigens challenge, and the increased release of viruses into the blood of number of CD cells decrease correlations with development of symptoms of AIDS. Maybe several. What about immunopathology? Why immune systems cannot eliminate these viruses? Neutralizing antibody which produce against glycoproteins 120 participate in antibody-dependent cellular cytotoxicity. Antibody-coated viruses in infections, and it can take up by macrophage. That's why it's, as I said, antibody is not effective. CD8 T cells or T killer cells generating the response of HIV infected cells produce suppressive factors that restrict viral replications. As a result, T killer cells to attack HIV infected CD4 cells and destroy these cells, also to add immunosuppressive. And these viruses, as we said, can immune escape because it's very variety by antigenic structure. It can survive in macrophage in lymphocyte, persistence in these cells, and as a result, it gives serious fatally immunodeficiency. Let's discuss several words about epidemiology. HIV is thought to be derived from simian lentiviruses. HIV-2 is similar with CAV. It's a virus which isolate from sooty mango base, West Africa. Monkey from West Africa. The simian lentivirus isolate from chimpanzee in similar to with HIV. That's why thinking that initial human infections maybe have occurred in Africa relatively recently. The migration of infected people on the sites after 1960 brought the viruses in population centers. AIDS was the first known in male homosexuals in the US in the late 1970s and early in 1980s. AIDS has spread in epidemic proportions through the populations. Now AIDS pandemic is a global phenomenon. The last statistic about 2020 years. 37.7 million of people globally were living with HIV and 1.5 million in 2020 years become newly infected with HIV. The region with the most serious affected is sub-Saharan Africa, where is a more spread HIV-2 and other variations of these viruses. But thinking that HIV-2 produce diseases similar to but less severe than AIDS, initiated by HIV-1. Then we should be said several. What about transmission of these infections? HIV is present in blood, semen, vaginal secretions, infected people. That's why viruses spread through the sexual contact and exposure to contaminated blood and blood products. Also, it can be transmitted parenterally to newborns. That who is group of risk or populations of the higher risk? It's a sexual active peoples, intravenous drug abuses, people receiving blood transfusion or organ transplant, hemophilics, newborns of HIV positive mothers, especially sexual in Africa, and healthcare workers that can have contracted with HIV infection from accidental needle sticks or cuts or through the exposure, broken skin, contaminated blood. Stomatology is also a group of risk people. What are clinical symptoms obtaining during the AIDS? Initial HIV infections, symptoms like on flu or influenza or infection mononucleases. It means maybe temperature, sometimes maybe rashes, aseptic meningitis, and it subsides after 2-3 weeks. The symptoms stem from immune responses, widespread infections of lymphoid cells. Then the initial symptomatic period of HIV infection is followed by period of asymptomatic infections or persistent generalized lymphadenopathy. This period, as I said, maybe last years, maybe 1 year, maybe 7 years. During this period, viruses replicate in lymph nodes. When the number of CD4 cells critically decreased, usually said that is less than 450 per milliliters, then latent period of HIV changed to AIDS. And on this period, registered opportunistic infections. When CD4 cells less than 200 per milliliters, it involves one of its more significant diseases as HIV wasting syndromes, weight loss and diarrhea more than 1 month. Kaposi sarcoma indicated diseases, it's vessel tumors. Pneumocystic pneumonia, a major things of AIDS. Mycobacteria avium intracellular complex infection and cytomegaloviral infection diseases. These diseases, how it's called, market or labeled diseases for HIV patients. Western syndromes, weight is lost, diarrhea prolongs the month, that's why it's a very severe symptomatical. Different opportunistic infections, fungi as aspergillosis in lungs, candidiasis on skin, oral cavity, trachea, bronchi, lungs, sepsis maybe. Kaposi sarcoma, it's vessels, tumors on the skin, also maybe in oral cavity. Oral hairy leukoplakia, it's AIDS marked infections. That's why all of these and other different opportunistic infections may be the reason of dire AIDS patients. Let's discuss laboratory diagnosis. Then, the recent infections or late-stage diseases can be indicated by the findings of the P24 viral antigens by ELISA test, viruses by culture, reverse transcriptase or viral RNA reverse transcriptase polymerase chain reactions, RT-PCR. This method now also used for detections of coronaviruses. And serological test. ELISA Western blot reef is used for document HIV infections, but this test cannot identify recently infected people. Then, if you talk about serological test, you should be know that HIV antibody may develop slowly, taking from 8 weeks, most of patients to 6 months, and more than for 5 courses. That's why we can detect an antibody only after 1 or more months. But for routine screening health, people usually use ELISA test for detections antibody against HIV. But ELISA can be fairly positive result. Then, for confirmed diagnosis, obligatory to repeat diagnosis with PCR or with Western blot analysis. Western blot analysis is a more specific procedure because it gives ability to determine antibody against different antigens. Antibody against glycoproteins 160, GP41, GP120, P24, P31. That's why Western blot assay is used for confirmed seropositive result. In some of laboratory, it's not, usually it's binding with scientific laboratory, can cultivation viruses. Viruses co-culture with patient's peripheral blood mononucleus cells with from cells from the donors with in presence of interleukin-2. Also can be used lymphoblastoid cell cultures as a MT4, Jucart and SIM. Then immunological studies also help to put diagnosis analysis of T cell subsets used for to determine status of HIV infections, that is count of level of CD4 cells, and especially compare number of CD4 lymphocyte and CD8 lymphocyte. Ratio of T helper to T cytotoxic lymphocytes, and for it's abnormally low for HIV infection people. That's why on this slide to show how to correlations number of viruses. Yes, we look on this by times in months. CD4 cells, yes, as on this. Antibody against HIV and how detection of antibody, especially if we use Western blot, give ability to detect stage of HIV, because if it's big amount of antibody against P24, that is very often means it's result, it means that latent infections change into acute AIDS. Let's discuss treatment. As I said, until this time, we cannot completely eliminate these viruses from infecting people, but use different antiviral drugs, antiretroviral drug, now which found we can prolong the life of these patients, prolong on very many years, and it's a very good and it's a very important, because it gives ability to chance these people, because maybe after some of years we found effective drugs or maybe vaccines which completely eliminate these viruses. Then, different target used for antiretroviral therapy. One of maybe first of target is reverse transcriptase. It means drug which act, this group of drugs calls reverse transcriptase inhibitor because it's act on enzymes reverse transcriptase. Another group of drugs, integrase inhibitors, which act on enzymes integrase. Next target, protease inhibitors, which stop to cleavage polypeptide and structural proteins. and maybe the best new generations of drug is a fusions or entry inhibitors, which don't give ability to fuse viruses with receptors on the surface of host cells. That's why anti-HIV drugs classify on nucleoside analogs, reverse transcriptase inhibitors, non-nucleoside reverse transcriptase inhibitors, protease inhibitors, integrase inhibitors, inhibitors of fusion and penetrations of the viruses. Nucleoside analogs reverse transcriptase inhibitor, they are phosphorylate bicellular enzymes, inhibit the reverse transcriptase, and after incorporation into DNA causes chain terminations. The more famous are zidotymidine, zidovudine, now dedoxycidine, zalcitabine, etc. The different commercial name. Next group, non-nucleoside reverse transcriptase inhibitor. They inhibit the reverse transcription by other mechanisms, usually by binding with active centers of enzymes. Example, nevirapine, delavirdine, efavirenz. Next group, protease inhibitors. They block the cleavage protease and as a result inhibition the cleavage of GAG and GAG-POL polyproteins. As a result, not formate structural proteins for new viruses. Examples, saquinavir, ritonavir, indinavir, etc. The next group, inhibitors, it's a maybe new generations of antiretroviral drugs, inhibitors of fusion and penetration of viruses. The first, infovertid or fuseon. This is drug block HIV from from getting inside healthy cells. Don't give to binding with receptors. Next, glycoproteins 120 attachment inhibitors. This is also a new class of drug with examples fostemsavir or ricobia. The target for these drugs, glycoproteins 120 on the surface of viruses. As a result, stopping from being able to attach itself of the CD4 cells. And results, viruses cannot attachment and penetrate in host cells. In other groups, CCR5 antagonists. The name of drug, Mabiroc. It's also stop HIV binding, but with another receptor, CCR5 receptors. Stop also step fusion and penetration viruses in cells. And if we talk about therapy, that is prolong the life of people. Therapy usually for prevent formation resistance for this drug, use not one type of drug, use cocktail or mixed from different group, from different mechanism of actions. For example, indinavir plus azidotamidine plus lamivudine. Inhibitors of reverse transcriptase, nucleoside, non-nucleoside, and inhibitors of protease. let's discuss several words about prevention. The first, educations, informations, monogamous relationships, the practical safe sex, use condoms, microbicidals, needles must be shared and intravenous drug abuses or reused in clinics. Blood and blood products should be screening on present these infections. If we talk about only general, method of prophylaxis, which usually used for blood transfer infections. Vaccines against this time, against HIV, is not available. If we talk about method of prophylaxis for medicine workers, special, and also for stomatology as group offering then gloves, protective clothes, mask, eyeglasses, special care should be taken, needles and sharp instruments. HIV contaminated materials can be disinfected with 10% bleach solutions or with ethanols or with another substances. Today that's all. Our lecture is finished. Goodbye.